Let's talk about the matrix now. The matrix is formed of a protein called osine. So osine is the protein that is present at the matrix of bones. The main salts found in the matrix are calcium phosphate, calcium carbonate, sodium chloride, and magnesium phosphate. So these are the main bone salts which are available in the bone matrix. Of these, calcium phosphate is maximum in the vertebrate bone. So calcium phosphate, that is the most important salt of vertebrate bone, as it is present in the largest quantity. The Haversian canals, that's a characteristic feature of the mammalian bones, are present in the matrix. So here we can see the Haversian canal. Each Haversian canal contains an artery, a vein, a lymph vessel, a nerve, and some bone cells. All are packed in with the connective tissue. The Haversian canals are interconnected by the transverse channels, which are known as the Walkman's canals. The matrix also has numerous inactive bone cells, which are the osteocytes. So osteoblasts are the cells which are important for bone formation, and osteocytes are moreover inactive. These are the inactive bone cells which are present in the bone matrix. So here we can find the osteocytes, their structure. The latter contain reduced number of cell organelles and often store glycogen. So glycogen, that is nothing but a polymer of glucose. Polymer of glucose, the stored form of glucose that is formed by the process of glycogenesis, that is going to be stored in the bones. An osteocyte is surrounded by a fluid-filled space, the bone lacuna, which leads into fine radiating channel. These are the canaliculi. So here we can observe the canaliculi, we can observe the osteon, the unit, Haversian canal, and definitely the osteocyte, which are the inactive bone cells. So osteocyte is surrounded by fluid-filled space, the bone lacuna, which we can understand by this diagram very clearly. And these are leading into fine radiating channel, which are the canaliculi or minute canal-like structures. So in the developing bone, the osteocytes give off several projections called protoplasmic processes or philopodia, which extend through the canaliculi. So protoplasmic processes are observed in the developing bone. Osteocytes, though are inactive cells, they give off several projections, which are philopodia. With the help of canaliculi and protoplasmic processes, one osteocyte is in contact with another osteocyte. So osteocytes are, though inactive, able to maintain good connection with each other. This is because they are forming canaliculi and many protoplasmic processes, and definitely these two are the spaces in between. Here we can find the osteocytes. They are capable of maintaining a coordination amongst each other. The Haversian canals are clearly visible. The unit is osteon, and these are the canaliculi, which are the protoplasmic processes extending from the osteocyte in a developing bone. The matrix of the bone occurs as layers called lamellae. So here we can find the lamellae. These are nothing but layer of matrix of the bones. Periosteum, the outer membranous layer, the Haversian canals which are present, the canaliculi, structures important for connection between the osteocytes, and here are the osteocyte cells inside the bone. The Haversian lamellae occur around the Haversian canals. A Haversian canal with its surrounding lamellae and osteocytes constitute a cylindrical unit of bone that is called Haversian system or osteon. So Haversian system is nothing but also known as an osteon. That's a unit, a cylindrical unit of bone. It is consisting of the Haversian canal and its surrounding lamellae and osteocytes. As a whole, the Haversian system is a functional unit of bone that is formed of lamellae, Haversian canal, and definitely the osteocytes, which are capable of maintaining themselves.